I'm Hans Andersson. I'm a clinical geneticist who works at the Hayward Genetics Center at Tulane School of Medicine. I've been working in genetic medicine for about 30 years, and I've witnessed an amazing evolution of a field from mostly a research area to becoming an essential part of medicine. I recently co-authored a book with Whitney Stewart called Genomics, A Revolution in Health and Disease Discovery. The book is written in a series of clinical cases and examples of the most recent practical applications of genetic testing to healthcare and ancestry. So much of what we're able to do today in clinical genetics is based on the Human Genome Project. It's hard to imagine now, but in the 1990s, it was only possible to sequence small pieces of DNA a few hundred base pairs long. By using many thousands of such sequences, created by hundreds of people, the Human Genome Project was able to string together a single, complete human sequence. We discovered that each of us carries 3.2 base pairs in each of our cells, which controls all aspects of growth and development, health, and sometimes disease. So if we're going to make use of those 3 billion base pairs to help people be healthy, we were going to need to be able to sequence far more rapidly than in the 1990s. The Human Genome Project led to the development of new techniques such as next-generation sequencing. With these new methods, one could sequence a person's entire genome in a matter of a few days. The challenge then becomes what to do with the vast quantities of information for which we have no prior understanding. We've learned so much in the last 10 years, but we continue to add to the information that allows us to interpret a person's DNA sequence for the purposes of health and ancestry. Because DNA sequencing has become so much less expensive, many commercial companies have made direct-to-consumer testing available for ancestry and human health traits. These tests use a person's saliva or cheek swab DNA to discover some limited DNA sequences. National Geographic started this approach in 2005 with an early ancestry testing program. They would sequence certain DNA fragments from your spit and tell you about the migration over thousands of years of your ancestors. In that way, they could tell you about your mother's family migration and your father's family migration. Because these early studies in the 2000s were done with limited database information about populations around the world, the results were preliminary. This is one of the biggest limitations we have in understanding human DNA sequences today, a lack of diversity in the DNA samples and limited understanding of what they do. It's like having a telephone book. Remember those? Without the names and addresses. Well, we're slowly filling in the names and addresses of the DNA sequence for humans. And as we do, there's a tremendous amount of practical information we can use to help diagnose rare disorders and identify health risks for people. But did you know that only about 2% of a human's whole genome contains genes for which code for proteins or have known functions? What's the other 98% of the genome doing? Well, if this subject interests you, I hope you'll read our book, Genomics, A Revolution in Healthcare and Disease Discovery. Many of these issues are discussed in the book in plain language and demonstrated by real cases. Genomic medicine is in its infancy and will certainly become an essential part of healthcare and medicine over the next decades.